day before, well, this will be our last content day for the exam. I'll go over that stuff in just a second. My boss, Dr. Lydiard, the chair of the department, asked me to pass this information along to my students. It's just about dates and classes. So let's see. The last time to register for a second 8-week class is tomorrow, this Friday, and then those classes begin Monday. So the second week classes, I don't know if you're familiar with those. I was not familiar with that concept when I started teaching here, but the second 8-week classes run for half a semester. So you can potentially, some courses like Spanish, I know they used to offer a Spanish 1 and 2 in one semester, and you just took 2 half semester classes. But anyway, the point of him announcing that, or telling us to announce it, is if you feel that you are doing so poorly in a class that you won't recover, and I'm not encouraging anyone to drop or anything, but this would be the time to make that very, very important choice if you need to drop a class, not necessarily this class, because you still have a chance. Now, there aren't second half biology classes. Some semesters there are, this semester there isn't, but there are other classes in other departments you could take just as a, to salvage your GPA and not make a choice that you can't reverse. So I'm not asking you or suggesting that you do it, I'm just putting that information out. That is a decision that you need to make on your own. I will say that the boss that hired me 3 bosses ago when I first started working here was super hardcore, and he taught this course because he wanted to scare students straight, like right out of the gates. He wanted people to be afraid of this subject. I don't know why. It's a very strange approach, very antagonistic. And so when he handed back his first exam, if people had done poorly, we used to drop classes using the little postcard. You filled it out and you took it to the registrar. This was pre-online stuff. And so if you did poorly on the exam, he would hand your exam back with the drop form as a hint. You might as well just give up now. It's really sad. Okay, so anyway, back to what I was discussing. So today is Thursday, and then we have Tuesday, then we have Thursday. This is our exam day, keep that in mind. And that's our last day before spring break. On Tuesday, we're going to review, and we're going to have the quiz about the aerobic respiration pathway. Today we're going to finish our material. We don't have a great deal of material, so I'm going to back up and review a couple topics that are important, and then we'll finish out the material, maybe get out a little bit early. The review is posted. Please take a look at this before Tuesday, so that on Tuesday, if you have questions or points on the review that you're not sure about or not comfortable about, we can touch on those and make sure that you are well prepared.
Is it the same as it was when you posted it a week ago?
I have not changed it since I posted it. And this test, I was looking over it before class just to get a concept if I had forgotten to say anything. And this test is 55 questions. I don't remember what the first one was. 45. So it's a little bit longer, but not much really. It won't take you any more time than the first did. Okay, so let's review a couple things, and then we will finish out our topic of aerobic respiration. To remind you some of the highlights, when you're studying this stuff, you really want to think about what goes in and what comes out of these reactions. What are the inputs and outputs, and what is gained each time? It's my favorite sound. Opening of a soda. At least I think it's a soda. Is it soda? What is? Don't even. Just keep it behind her so I can't see it. Okay, anyway. So we have our 4 reactions: glycolysis, pyruvate breakdown, citric acid cycle, oxidative phosphorylation. Actually 4 stages. Each of those stages has multiple reactions. The first stage, we're trying to take in an organic molecule, which in this case, the one we focused on is glucose, and we're trying to break that into 2 smaller molecules. So we go from the 6-carbon sugar to 2 3-carbon molecules. We yield 2 ATP and 2 NADH. Do you remember how many we put in? 2. So we invest 2, we actually make 4, and then we get 2 back. So we end up with an excess of 2, but we do have to make an investment to get there. Okay, then we take our 3-carbon molecule and we head into the mitochondria. And so in the mitochondria, the pyruvate breakdown happens. The pyruvate is transported into the matrix, and then it is converted by an enzyme into an acetyl group, which means we're going from 3 carbons to 2. So that 3rd carbon has to go somewhere, and the somewhere that it goes is into carbon dioxide gas that can be released by the cell. We don't get a lot out of this, but we do yield 1 NADH for each of the 2 pyruvates that went in. So it's not too bad. The 3rd stage, we really do generate a lot. It may not seem like it because only a couple ATP are made for each, or 1 ATP per acetyl, so 2 total. That doesn't seem like a lot, but the NADH and FADH2, that's where the action is. That's where all the power is going to come from later. So the acetyl group becomes carbon dioxide, so now all the carbons that we took up in the first place are gone, and the electrons from the bonds that were holding those carbons together are now stored in our electron carriers NADH and FADH2. And then the final cash out, which we did on Tuesday, was to convert the NADH and FADH2 using an electrochemical gradient into ATP. And that process had a name that you'll want to know and remember, which is chemiosmosis. Okay, chemiosmosis is when we use an electrochemical gradient to generate ATP. All right, I'm going to skip through a couple things here. Here is a graphic version of the thing that we just discussed. I'm sure that you've already memorized this. You've probably made 100 copies and been doing this in all of your free time because that is your ticket to the perfect quiz on Tuesday. It'll be your finest 5 minutes of the semester. And so one thing to remember about this illustration or graph or whatever you want to describe it is that when we have a molecule that has descendants, that molecule can produce, in this case, 2 pyruvates. Because glycolysis also makes NADH and ATP, we'll put those here. Our ATP always goes on the dotted line. That's just an easy way to visualize where ATP is being made. And then nothing else becomes of this NADH until the end, but the pyruvate has more descendants, right? So the pyruvate yields acetyl-CoA, carbon dioxide, and some of the electrons from pyruvate go into NADH. All right, so again, carbon dioxide and NADH, those are going to be used elsewhere or just leave the cell in the case of carbon dioxide, but acetyl-CoA will have more descendants. So our arrow comes down from that one only, and we get carbon dioxide, more electron carriers, and ATP. And then in the end, all of the NADHs from all over the chart, as well as the FADH2s, go into oxidative phosphorylation. So know this. Do we know this? How close are we to knowing this? Very close? Oh, 1 thumbs up. The energy drink is kicking in. Okay. Theoretically, the energy drink is kicking in. Okay, so a couple things about glycolysis. Again, just trying to hit the highlights so you'll remember these points of emphasis. We input 2 ATP, and there's a reason that we are phosphorylating this glucose to add these ATP, or to add the phosphates from the ATP. Of course, once we add 2 phosphates, it's no longer glucose, it is now fructose bisphosphate. But when the glucose is phosphorylated, there's a reason. Does anyone remember the reason we do that? Yes?
So it can't come back out?
So it can't come back out. So there's a little trick. Actually, there's two things. That's the main thing. And the second thing is related to that. When the glucose goes in, I'll just abbreviate it with G. If it were to go across the membrane like this, then it would seem like the concentration of glucose is then higher on the inside than outside. Okay? But the trick that the cell plays by phosphorylating this, there are 2 outcomes. The first outcome, as we said, or as you said, was that the glucose now can't go back out the door that it came in. It's shaped differently, it's larger, it's stuck inside. But the other difference is now it doesn't look like there's more glucose inside than out to the cell because the cell no longer acknowledges this as glucose. So if we have, let me make it more clear like this. If we had 3 glucoses outside the cell and 3 glucoses with phosphate inside the cell, as far as the glucose concentration goes, these don't factor into that because they're not glucoses anymore. And so by converting it to glucose 6-phosphate, the glucose can't leave and the cell keeps thinking, okay, we're going to keep transporting more of these into the cell because we don't have enough. So it's a little mind game, you might say, that the cell is playing, or the glucose is playing on the cell. Okay, and once we add those phosphates, we create unstable bonds that can then dedicate or donate these phosphates to ADP to make ATP. So we call that, substrate level phosphorylation. So these unstable bonds are ready to break. And when they break, they're going to release that phosphate. If there's an ADP waiting for that, now we can generate ATP. The same thing happens to this unstable high energy phosphate bond. It comes off and adds to the ADP to make energy. Okay, the main point of this diagram, other than just to point out what all the things are, is to show where things are happening. So if I were to say which or where in the cell does glycolysis happen, you would say the just glycolysis, not the mitochondria.
Plasma membrane.
Well, the plasma membrane would be around all of this. What's inside the plasma membrane but outside of the mitochondria? The cytosol. So glycolysis is the only reaction or the only set of reactions we've talked about that is not mitochondrial. Okay, so that's an important point. It happens in the cytosol, and then everything else happens in the mitochondria. Okay, so that's a key thing. What other highlights do I want to hit before we finish out? Okay, this one. The last phase that we finished off on Tuesday was oxidative phosphorylation, and a few key points to remind you about this. Outside of the fine details, let's just hit the highlights. One is that we are now using all of the high energy electrons that NADHs and FADH2s brought into this equation. Okay, so we've been collecting those since glycolysis happened. And those NADHs and FADH2s with their high energy electrons are ready to do what electron carriers do, and that is give those electrons back. And so they're going to donate it to these things which are going to transport protons across the membrane, okay? So the energy from the electron is doing the work, the NADH is just donating the electron, and the electron actually is carried through the membrane by different electron carriers, not FADH2 and NADH, but different electron carriers like ubiquinone and cytochrome C. As it travels through the membrane, that energy is used to do work, okay? And the work it does is create this proton gradient, all right? So these protons are being pumped against their concentration gradient, which requires energy. That energy comes from the electron, and this is the concept of chemiosmosis, using an electrochemical gradient to power the synthesis of ATP. And it does so with that enzyme, which is called ATP synthase. So this enzyme is going to revisit us when we talk about photosynthesis after spring break. But this enzyme uses the energy from hydrogen ions traveling back down their gradient to restore that equilibrium, and that energy actually physically cranks that ATP synthase enzyme so that it can then use that mechanical energy to add a phosphate to ADP. So this is the result of all that electron energy boils down to this enzyme powered by the electrochemical gradient, which is, again, the process as a whole is called chemiosmosis. Okay, the other thing to mention about this is this little section right here, which looks like just a minor blip, but we want to zoom in on that for just a second, because this is kind of the whole point of aerobic respiration, is that in order for this to work, we need oxygen. In the absence of oxygen, none of the stuff we've talked about happens except glycolysis, and we'll highlight that in just a second, then we'll hit our new material. But this oxygen gas, which again is distributed to our cells from our blood, and it's in our blood because we breathe it into our lungs, and our blood cells take it into tissues and release the oxygen so that cells can utilize it to make energy. And the reason that you need the oxygen is that at the end of this electron's long journey, each electron's long journey, it has to go somewhere. And oxygen was called the final electron acceptor, okay? And the final electron acceptor takes that electron, adds it to the oxygen, grabs a couple of hydrogen ions, and produces water. And so this, remember the overall reaction of aerobic respiration that we want to think about also as foreshadowing for photosynthesis is that we have glucose plus oxygen produces carbon dioxide plus ATP plus water. So that is aerobic respiration. And if we go in the other direction, using carbon dioxide and ATP and water and light energy, we can produce organic molecules like sugars and oxygen. Well, we can't, plants can. Okay, so that is that. Then we did this again. And we talked about briefly the way that we keep our energy reserves. And the reason I mention that is because it's part of the idea that we're not just eating sugar, right? We've acted in this whole section like glucose is the only way that we get energy, but we actually get energy from lots of different things, and we need to do that because that's how we store our energy long term. So if we couldn't break down fats and put them into this process, we would be in trouble. We can't just live on sugar and energy drinks alone. So I got that in there. Okay, I'm not going to worry about this slide. You don't need to know that. And then the very last thing we did, I believe, was this. Well, I couldn't put them both up there, which was anaerobic respiration. In anaerobic respiration, we do not have the final electron acceptor of oxygen. So energy production can continue, but it is severely impaired because we don't get to utilize all the NADHs down the line as pyruvate is broken down into acetyl-CoA and then into carbon dioxide. So all those electrons from the sugar molecule, we only get to harvest the ones in the first stage. And so pyruvate is still made. That pyruvate is then converted into lactate, right? And the lactate then, it says secreted from the cell, but that lactate does cause discomfort. As you know, if you've ever exerted your energy so much that you've run out of oxygen in tissues, you have that burning sensation. And now eventually the lactate is removed, and that's one reason people massage athletes who are having cramps in their muscles, because it gets blood flowing and that removes some of the lactic acid. But anyway, all we get is the ATP. So the reaction doesn't proceed past that if we don't have oxygen waiting at the end. Now, if we think about some other organisms, not animal cells, but organisms like bacteria and yeast, they can also do anaerobic respiration, but they sometimes will produce ethanol instead of lactate. So they have a different process, and that ethanol, in the normal circumstance, can be secreted from the cell. In a consumer world, we actually, of course, harvest this technology to produce alcoholic beverages or fermented foods are made by this process when we allow things to actually undergo anaerobic fermentation, like sauerkraut, for example, is a good fermented food. But anyway, without oxygen, all is not lost, but most is lost. Right? Most of the energy you should be getting from that sugar is going to waste. Okay, let's finish out our material. Now we have a few items to discuss, mostly about the regulation of how we take up glucose in the first place, and then a little information about when things go wrong with our glucose uptake, which is to say diabetes. So we'll look at some of the kind of general facts about diabetes. We won't go into a full physiology type course, but one thing that we want to consider is that the cell does not just absorb glucose because it's there. The cell has to be told to, well, let me just demonstrate. In order for the cell to absorb the glucose that is in the blood, it needs to be told that the glucose is there, and that happens by sending a signal, which is a hormone signal, and a hormone signal in this case, of course, is going to be insulin. And when insulin is received by the cell then takes the glucose transporters that it already has often made, like they exist inside the cell, they're just not expressed at the membrane. And so upon insulin signaling, those glucose transporters go to the membrane so that the cell can then start to bring in that sugar. In the absence of insulin, those membrane receptors are not going to be there, okay? So the insulin is coming from our pancreas, and one of the things that is unique about the endocrine system is that the hormones can be made anywhere, right? We have hormones that are made in the pancreas, we have hormones that are made in other glands. A lot of hormones are made and components of our brain, but that doesn't mean they're going to be acting on our brain cells, just like the pancreas is not the place where we absorb sugar, right? We absorb sugar in our digestive system, but the pancreas is sending that hormone into the bloodstream, and this is the key part of the endocrine system. No matter where the origin of that hormone is, once it gets into the bloodstream, as long as there is a cell somewhere that has the right receptor, it can receive that signal. So the cells that receive the hormone insulin, of course, are at great distances from the source of that insulin, but once it's traveling through the bloodstream, if they have the right receptor, it doesn't matter where they are. They're going to be able to absorb that insulin, and then they will produce the glucose transporters, and they'll absorb the glucose. Okay, so I made a little flow chart, which you may have if you would download the slides, to show how these things interact. And so if we start out with glucose on our right side there, and our pancreas, specifically the beta cells in the pancreas, secrete insulin. Glucose is going to be absorbed by the cells, and then we'll go through this process that we have just spent 3 painstaking lectures talking about. Okay? But as we've said before, sometimes you have more glucose than you need in your blood, right? Oftentimes you do. And so if that's the case, we can save some of it. And so if we want to convert that to glycogen, right? And our glycogen can be stored in the liver. It also can be stored to some degree in our muscle cells. I don't have that up there, but both muscles and the liver can store glycogen. This is the polysaccharide we looked at when we were talking about macromolecules a few weeks ago. And this is our short-term energy reserves. Remember, just a very, very small percentage of our of our energy comes from glycogen. But nonetheless, we still use it a lot because it's so easy to make and then return to the state of glucose. So in order to convert it back to glucose, let's say you save some of that energy in the form of glycogen and you need to harvest it, but you're not having a meal, you're not getting any input through your digestive tract. Instead, you need to get that from the liver or the muscle. You secrete a different hormone, which is called glucagon. And that glucagon causes the liver and muscle cells to to break down the glycogen and release that back into the blood so that you can then have energy even though you haven't consumed any more of your meal or whatever snack. So the interplay of insulin and glucagon, they're both doing the work of getting glucose to be converted eventually to energy, but the glucagon is releasing it from the liver and breaking it down into glucose, and then the insulin tells the cells it's time to take this up. So you want to be comfortable with those two, the roles of those two things. Okay, the last thing then I want to talk about, have a few slides about diabetes. This is, of course, the failure of our body to properly regulate glucose intake, specifically from the blood. So let's look at a couple little sort of general details. You may already know some of this or be familiar with this. It'd be unlikely that you did not know or encounter or have a family member or maybe yourself even that has diabetes. It's so very common in humans. But not many people know a lot about it. If it doesn't affect you directly, maybe you've never thought about it. But it is a collection of diseases, so it's not just one specific problem. The idea is there's excess glucose, but there are different ways that you can be affected by this. Many of the issues that occur are unpleasant and yield things that you don't want. For one thing, increased thirst, unintended weight loss, fatigue. Your vision is heavily influenced by high blood sugar, and then you have an increased risk for infections. And of course, diabetes is not restricted to humans, right? Animals can have diabetes. In fact, it affects a lot of domestic animals. I've had a cat that had diabetes, and some cats are more prone to it than others, and dogs can have it as well. So this is not just a human issue. Bless you. The different types then, the first type that is fairly common is prediabetes. I'll describe what that is in a second. The second type that only occurs during pregnancy is gestational diabetes. The third type is called type 1 diabetes. This is also sometimes called juvenile diabetes. And then the fourth one is type 2 diabetes, and the M is diabetes mellitus. This is in your does anyone have the Blackboard slides available to them? This is in there, right?
I downloaded at the beginning of the year, so no.
It's not in there?
No.
Okay, I'll post them later than I must have just put it on my flash drive and not uploaded it to the thing. Okay. Well, I'll post it, so if you miss something or if you don't copy everything down, don't stress about it. I'll put it up right after class. I thought I put it up, I guess I didn't. Okay, so let's talk about what it means to have diabetes. What is the diagnosis? So the diagnosis is all about blood sugar that is usually taken as or that test is usually run on an individual that has fasted. So to fast for a blood sugar test, usually you're told to not eat or drink anything for 8 hours. And then they take your blood and they look at the amount of glucose in there. And what they're looking for is, in the absence of food, how much glucose does your body maintain in the blood? And there's a healthy amount and an unhealthy amount. And so it says normal, this would be best case scenario, you would have less than 100 milligrams per deciliter. That's what that stands for. But if you have pre-diabetes, that means you are above 100, but less than 126. And these numbers seem just like they're pulled out of the air, and actually they change over time. So at one point, it was okay to have sugar at 105. Then people realized that was too high. People with 105 blood sugar levels were actually developing diabetes later, so they changed the standard down. Just like with your cholesterol, sometimes that number changes. People decide after, long-term health studies that maybe the numbers they had weren't so great after all, and they changed the standard. But for now, this is current. Less than 100 is the most desirable. A little over 100, but not quite to 126, is considered pre-diabetic. And pre-diabetes is usually not treated by medication. It is usually, what will usually happen is your doctor will encourage you to try and change habits, to try and do something in your lifestyle that would change that. Sugar is one big risk factor. I'm not sure why red meat is a risk factor. That I don't know, but sugar obviously makes sense. Exercise will sometimes reverse pre-diabetes in people that otherwise were not very active. So there's some things you can control and some things you can't. Not all aspects of diabetes are under your control, but some are. And so certainly your diet is one of those things you can control. The primary risk, but not the only one, would be obesity or being overweight. Another huge risk factor for diabetes is your genetics. So, if you have a family history of diabetes, you would want to be extra vigilant to monitor your blood sugar so that you could, as soon as you knew that you might have an issue, you could take action to improve your health, hopefully. Okay, gestational diabetes. This is a really unique situation because it only affects females during pregnancy. An interesting thing about gestational diabetes is that after the pregnancy, usually it goes away. During the pregnancy, there are things you can do to move the needle in your favor. One thing is to have a really low sugar diet, which of course is really hard during pregnancy, as you can imagine, to remove certain foods from your diet. There's some medication you can take, but of course, as a pregnant mom, you have to limit what types of medication are safe for you and the baby. And then exercise, of course, there's never a bad time to exercise. Exercise is always good. And a long-term effect of gestational diabetes is that your risk for type 2 diabetes, which develops for most people later in life, goes up. So if you had gestational diabetes and everything returns to normal after the baby's born, you still want to, I'll use the same word I used before. You want to be vigilant about monitoring your health because you've moved into a different risk category. Doesn't mean you'll get it, but it means you want to be on the ball monitoring that. Okay, and then the type 1 diabetes, this would be often, again, called juvenile diabetes. This usually is detected early in childhood or the teenage years. Some of the complications that happen from juvenile diabetes or type 1 diabetes mellitus are cardiovascular issues. Okay, having high blood sugar really does a lot of bad things to your circulatory system. So those are things, of course, you have some control over treating these types of issues with medication and lifestyle. Nerve damage. One issue that often affects people who have type 1 diabetes is vision. Their vision is impaired. And then because of the circulatory issues, many people with type 1 diabetes have foot issues because the way that the blood will pool at the bottom of your body if your circulation is not working as it can. Now, the reason the type 1 diabetes happens is usually, well, I think it's safe to say, is never related to lifestyle. And so type 1 diabetes is an example of an autoimmune disease. And if you're not familiar with that concept of autoimmunity, this is when your immune system attacks your own cells. And there are lots of different autoimmune diseases. One autoimmune disease that affects a large number of individuals, and disproportionately females, is lupus, systemic lupus erythematosus. You may have heard of that. That's an autoimmune disease where your body actually makes antibodies against your nuclei and your DNA, and it causes a lot of different things, including joint pain, and it causes a rash on the face sometimes that makes it look like you have the mask on a wolf. That's where the name lupus comes from. That's the species name for that canine. In any case, that's another example of an autoimmune disease, but this particular autoimmune disease happens when your body starts to attack the cells in the pancreas that secrete insulin. And it's not always clear, I don't think it's ever clear why your body starts to do this, but something fails in your immune system and you don't recognize those beta cells as your own cells, and your body starts to kill them. And then, of course, if the beta cells go, then you don't make insulin. If you don't make insulin, then you can't absorb the sugar from your blood, and that's why you have to then treat yourself with insulin shots generally. Okay, and then type 2 diabetes. This can, as it says, occur at any time during life, but it's almost always later in life. And typically someone that gets type 2 diabetes will have a period of pre-diabetes before it morphs into that final stage. And like the other issues with diabetes, anytime you have high blood sugar, you're going to have some of the same things that you had in the other types of diabetes too. Increased thirst and urination, increased hunger, but you're still losing weight because you're not absorbing those nutrients, and then frequent infections. And again, because of circulatory issues, your extremities are often affected. In fact, just as a little anecdote, do we know what the word anecdote means? There's a reason I'm asking this. Does anyone know, is anyone willing to say what is meant by the word anecdote?
Isn't it just a personal story?
That is the best definition anyone has ever given. It is a personal story. The reason that matters is that, the reason that matters is people often use it wrong. And wait, make sure I'm spelling this right. I just want you to enjoy this one moment with me, but I gotta make sure I'm spelling right. Yes. So what does this word mean?
Like a cure.
Like the cure. You know so much. You a good speller?
I'm dyslexic.
Wow. Okay, here we go. Anyway, the reason I mention this, there's a reason I'm going to use the word anecdote. But if you are, the antidote is like the cure for something. And, with all this political furor and everyone's angry at everyone else, there was a protest. I don't even remember what the protest was for. But somebody was saying, we want to be the antidote for this problem. But instead, all the signs said, we are the anecdote, which makes no sense, right? It's like saying, we are the personal story to solve this problem. And they were so angry, but it was hard to take them seriously because they were using a completely wrong word. Doesn't have anywhere close to the same meaning. But anecdote is a personal story. And one of the things that in science, hold on a second, make sure I'm spelling this right. One of the things that people often say in science is that we have anecdotal evidence. And this is an important thing to think about because anecdotal evidence means you have had an experience that you are relating in order to support an idea. So for example, I have anecdotal evidence that, I'm just gonna make something up, that I run faster if I have a honey bun before a 5K. That has been my experience. Surely that honey bun is not helping me run 5K. It's probably weighing me down with all of that lard and fat. But that's my personal experience. So I could say anecdotally, the best way to run faster during a road race is to have a honey bun first. But it's not really true, right? It could be true, but it's just my experience. And so what I was gonna say is my anecdotal experience with animals that have diabetes, if you happen, I'm sure most all of you have pets or will someday, was this increased thirst, which is that a cat who was older and I later learned had diabetes, could not stop drinking. You got out of the shower, it jumped in and licked the shower dry. It tried to get into the toilet to drink the water. If there was a sink running, it would jump up and try and drink it. So it was almost like a vampire situation. It could not help itself. And the second anecdotal experience of a diabetic animal was that there were fruit flies in the litter box. Why would that be?
The high blood sugar.
The sugar, yeah. The urine smelled sweet because it was full of sugar. And so fruit flies would go to the litter box, which is uncommon, right? Usually fruit flies look for food. So anyway, that's my anecdotal experiences with cats and diabetes. Unfortunately, I did not have an antidote. I did give shots for a while, but it was too late, unfortunately. R.I.P. Isabel. Okay. Do I have anything else? Oh, blurred vision. Okay. So why does type 2 occur? So type 2 is not an autoimmune disorder where your body attacks itself. Okay. Instead, type 2 diabetes happens because over time, one of two things takes place. Either the cells no longer respond to the insulin, right? So you're still making the insulin, but your cells aren't reacting to it. And that's really a difficult thing to deal with because if you are not producing insulin, you can always add more, right? You can take an insulin shot. You can see how much insulin you need and give it to yourself. But if your cells are insensitive to it, then all the insulin in the world is not going to help. So that's one issue. The second one is that the beta cells, they're not dying, they're not being attacked by your immune system, but they're just not making enough insulin anymore. So for whatever reason, there could be a number of reasons for that. You are just not producing the insulin you need, even though your cells are able to respond to that if they get it, you're just not making it anymore. And so that's a case where you actually can, medicate yourself with insulin and resolve that situation. Dum, dum. A look into my soul. Just darkness. Okay, that's it. I'm done. Go for it. On Tuesday.